Hello students, welcome to BioDigest. We are actually dealing with the composition of the blood. Already we have discussed the liquid portion of the blood, that is blood plasma, in my previous video. In that video, the cellular elements, that is RBC, WBC and blood platelets, of that RBC was also discussed. So in this particular video, we will learn about the WBCs. WBCs, otherwise white blood cells, or called as leukocytes. So this is coming under cellular elements. I told you RBC was discussed in my previous video. Please do watch it. So WBC. Question can come for the board exam. What are the structural differences between an RBC and a WBC? So how will you write this answer? RBC, what is the shape? Already I have explained, RBCs are biconcave disc-like structures. Okay, I'm lightly shading in the middle. What about the structure of a WBC? WBCs are actually amoeboid structures. Then RBCs, they lack nucleus. I told you that when the RBCs are formed, they are actually having nucleus, but when the RBCs become mature, they become enucleated. Okay, but WBCs, they will be usually having a nucleus. Okay, so we have seen the structural differences now. So RBCs, they are biconcave disc-like structures. WBCs are amoeboid in shape. RBCs lack nucleus, or you can write RBCs are enucleated. WBCs possesses nucleus. Then RBCs, usually they contain a respiratory pigment called as hemoglobin. In its colorless spongy stroma. So respiratory pigment called as hemoglobin is present in RBC, whereas the respiratory pigment hemoglobin is absent in WBC. So hope the structural differences between RBCs and WBCs is clear to everyone. Now, WBCs, usually I told you they are amoeboid in shape. So imagine that this is a capillary. Okay. So WBC is having one peculiar nature. So this is the capillary. Capillaries are very thin. Okay. For showing it clearly, I am drawing little bigger diagram. Imagine that this is a WBC. Okay. It's having a nucleus. So by producing pseudopodia, pseudopodia means false foot, like this. Okay, this is pseudopodia. A false foot is produced. By producing the pseudopodia, they are actually able to squeeze out through fine narrow capillaries, okay, like this. So I'll draw the next diagram, as a result of which what will happen? Okay, they are trying to move out through this capillaries, and finally this WBCs, they will be squeezing out like this by producing the pseudopodia, okay, and they will be moving out. They will come out of this capillaries this, the irregular. Okay, so I have shown this various diagrams. What is this process called as? This process is what is called as diapedesis. Again, this can come for the board exam. What is diapedesis? Diapedesis is the squeezing out of WBCs by producing pseudopodia through fine narrow capillaries. So squeezing out of WBCs through fine narrow capillaries by producing pseudopodia is what is called as diapedesis. Okay, so that is one peculiar feature that is carried out by the WBC. Now coming to the two major categories of WBC. One is granular WBCs, otherwise called as granulocytes. The second category is non-granular WBC, otherwise called as agranulocytes. The name itself is giving some idea. What are granular and agranular WBCs? Or what are granulocytes and agranulocytes? Granular WBCs means usually the WBCs will be possessing granules in their cytoplasm. Okay, I am not showing the nucleus here. Nucleus is of course present. So these are the granules that is present along with the other structures in the cytoplasm. Okay, you can see large sized grains. So if granules are present, such WBCs will be coming under the category of granular WBCs. Then what about non-granular WBCs? In non-granular WBCs, these granules will be absent. Okay, only cytoplasm, you can see the cytoplasm. But granules will be absent. So granular WBCs, which are they? Neutrophils, basophils, and eosinophils. So there are three granular WBCs. Which are they? Neutrophils, eosinophils and basophils. Coming to neutrophils. What is the peculiarity of a neutrophil? Okay, so neutrophil, these WBCs I told you they are amoeboid in shape. But the specialty is the nucleus will be 3 to 4 lobed usually. I am showing 3 lobes like this. Okay, so 3 lobed nucleus. You can see 3 lobes. 3 or 4 lobes like this. Then I told you cytoplasm. Then in addition to that, granules will be there. That is why this is coming in granular WBCs. Okay. Now neutrophils. Usually the granules will be staining with neutral dyes. That is why we are calling it as neutrophils. So neutrophils are WBCs where granules are present in the cytoplasm. The nucleus is actually constricted into 3 to 4 lobes. Then they usually stain with the neutral dyes. That means the granules take up the neutral stains. Then where are these WBCs produced? All these WBCs, that is the granular WBCs, are produced in the bone marrow. What is the function of neutrophil? Neutrophil is having a particular function that it carries out. It will be engulfing solid particles. Okay, this is the spelling. Engulfing solid particles, especially disease-causing germs like bacteria. That means it will swallow that. Okay, that is the meaning of engulfing this particles. Solid particles will be engulfed especially by neutrophils. Which type of solid particles? The disease-causing germs, especially bacteria. The process is having a name. It is what is known as phagocytosis. This again is an ICSE board exam question. Define phagocytosis. So how will you write the answer? Phagocytosis is the process by which WBCs, especially neutrophils, will engulf solid particle-like substances, especially bacteria. Okay, that process is what is known as phagocytosis. Now coming to basophils. Basophils again, this is, I told you, amoeboid. Then granules are there. Okay, don't forget to show all this. All these are the structures. That is common to everything. Only thing is that the nucleus will be having some difference. This is indistinctly lobed. Okay, so that is about basophil. Indistinctly lobed. Hope you can see. So indistinctly lobed means it is basophils. So the granules will be taking up basic stain, that is methylene blue. Okay, there is a stain called as methylene blue. So this granules is having the capacity to take up that basic stain, so called as basophils. Okay, so what are the features? Basophils, indistinctly lobed. Granules are present in the cytoplasm. All that is why it is coming under granular WBCs. Then they usually take up basic stain. That is, example of basic stain is methylene blue. Now what is the function that is carried out by a basophil? So basophils will produce a chemical substance called as histamine. They will release this into the bloodstream, thereby dilating the blood vessels. So that is the function that is carried out by basophils. Now coming to eosinophils. Eosinophils are actually WBCs which are two-lobed. Okay. Then granules are there, of course, in the cytoplasm. Then these granules will be taking up acidic stain. Okay, especially eosin stain. So these are eosinophils. Okay, so that is about eosinophil. Eosinophils, two-lobed structures. Granules are present in the cytoplasm. The granules will be taking up acidic stain, that is especially eosin stain. What is the function of an eosinophil? Again, it also carries out phagocytosis. Phagocytosis, that means engulfing disease-causing particles, especially bacteria. That is about granular WBCs or granulocytes. Now coming to the non-granular or agranulocytes. Two are there. Okay, we will see what are they. One is lymphocytes. Another is monocyte. So lymphocytes and monocytes are coming under the non-granular WBCs or agranulocytes. Now coming to lymphocytes. that is the non-granular WBC. This is the smallest of the WBCs. Okay. So usually it will be having a single large nucleus like this. I told you this is coming under agranulocytes. Okay, so you don't have to show any granules in the cytoplasm. Simply show the cytoplasm like this, small dots. No granules. So smallest of the WBC, which is that? It is lymphocyte. Where are these lymphocytes produced? Lymphocytes are actually produced again in the bone marrow as well as in the lymph glands like spleen, tonsils, etc. Okay, so lymphocytes, they are the smallest of the WBCs with a single large nucleus. Granules are absent in the cytoplasm. They are produced in the bone marrow and in the lymph glands like a spleen and tonsils. What is the function? Again, ingesting germs or phagocytosis. Now coming to monocyte. Monocytes, they are having a peculiarity. The cytoplasm, I am showing the cytoplasm. They are having kidney-shaped nucleus or bean-shaped nucleus. Okay. So if kidney-shaped nucleus is shown, that means it is a monocyte. Kidney-shaped nucleus, that is the peculiarity of monocyte. Now monocyte, at the site of an infection or an injury, the monocyte can convert itself into a macrophage. And they can ingest germs. WBCs, where are they produced? They are actually produced in the bone marrow as well as in the lymph glands like spleen, tonsils, etc. What is the lifespan of a WBC? Usually, the lifespan of a WBC is almost 2 weeks. But neutrophils will live only for few hours, after which they will be getting destroyed in the liver or in the spleen. So the destruction of the WBC is actually happening in the liver or in the spleen just like an RBC. When the number of WBCs increases manifold at the cost of RBCs, the condition is what is called as leukemia. Leukemia, commonly we will call this as blood cancer. So usually the number of WBCs will be increased. Okay, the condition, usually this can happen even when the body is having some sort of infections. But its count has to come down. Okay, because when we are having some sort of infection, we will be definitely taking the medicine for that. So after taking the medicine, it has to come down. So if the number of WBCs is not coming down, it is continuously increasing or it is going on in that stable manner, increased manner, then another test has to be conducted whether to detect whether the person is having leukemia or not. So this condition is what is called as leukemia. Now coming to the third cellular element of the blood, that is blood platelets. Blood platelets are otherwise called as initiators of blood clotting. Okay, they are actually responsible for the clotting of the blood. So blood platelets, usually they are minute oval shaped or circular shaped structures which will be usually found floating in the blood. They are non-nucleated. They lack nucleus. So this is how we have to draw blood platelets. Okay, minute oval-shaped structures like this. This is what is called as a blood platelet. They are non-nucleated. Megakaryocytes are some giant cells present in the bone marrow. Okay, so usually this blood platelets are actually budded off from this megakaryocytes. I told you they are called as the initiators of blood clotting. At the site of injury, the blood platelets disintegrate to release a chemical substance called as thrombokinase, which actually brings about the process of blood clotting. That is why these are called as the initiators of blood clotting. What is the lifespan of a blood platelet? The average lifespan is almost 3 to 5 days, after which again they are destroyed in the liver or in the spleen. Let us recall. White blood cells or leukocytes. What are the structural differences between a RBC and a WBC? RBC shape, biconcave disc-like structure. WBC shape, amoeboid. RBC, they are enucleated and they contain hemoglobin, the respiratory pigment. WBC, they have nucleus and they lack hemoglobin. The two major categories of WBCs: granular WBCs and non-granular WBCs. The granular WBCs include neutrophils, eosinophils and basophils. The non-granular WBCs include lymphocytes and monocytes. The smallest of the WBC is lymphocyte. What are the functions of leukocytes or WBCs? Phagocytosis. WBCs engulf particle-like solid substances, especially bacteria. The process is called phagocytosis. This is a defensive mechanism against disease germs. Inflammation. Inflammation occurs due to the reaction of tissues to injury and to localized invasion of germs. Formation of antibodies. The WBCs produce antibodies which kill or neutralize the germs or the poisons from them. Antibodies circulate free in the blood plasma. The antibodies are specific to the toxins for which they are produced. The antibodies act as antitoxins, which neutralize the poisonous effect of the toxins. Origin and life of WBCs. The WBCs are produced in the red bone marrow, lymph node, and sometimes even in liver and the spleen. Their average life is about 2 weeks, whereas the neutrophils live for only a few hours. What is leukemia? It is a cancer of the tissue forming WBCs whose number increases manifold at the cost of RBCs. Leukopenia. It is the abnormal decrease in the number of WBCs. Blood platelets, known as the initiators of clotting of blood, also called as thrombocytes. These are minute oval structures, non-nucleated, floating in the blood. These are actually budded off from the megakaryocytes in the red bone marrow. Their lifespan is 3 to 5 days and are destroyed mainly in the spleen. They are very important in the clotting of blood. Please do subscribe Bio Digest. Thank you.